Hello, everyone. Today I'll be presenting on some of our AMR wastewater work that we've been doing in especially low resource communities here in the state of Kentucky. And so like a lot of other labs, we began our wastewater-based epidemiology projects here on the University of Kentucky campus, where we used a dual-phase testing program to track positive wastewater signals to very specific buildings on campus, where we could then go in with more accurate testing modalities to identify specific students who were infected. And this helped us to potentially prevent a couple outbreaks where students either didn't know they were sick or were pre-symptomatic. So we were very excited to do that. And then next, we looked at where this data would be more useful and where it wasn't being collected. And we came up with rural counties on the eastern side of the state where there are plenty of data suggesting that people are getting sick, but we don't really know how the pandemic is spreading. And so we quickly learned that walking across campus to collect wastewater is very different than asking someone to ship it through UPS or things like that. And so we ran into temperature-related degradation and things like that. And so we really needed a extraction process that we could do very quickly that would sort of limit our travel time and do it at a cost price point that was conducive to a partnership with some of these more low resource communities. And what we came up with was a BSL-2 van that we outfitted. So inside of this 2000 transit van, we have everything that we need to collect, concentrate, and analyze via PCR wastewater for all sorts of things. And this really brought down our time to result from multiple days from shipping to sometimes as quick as a couple hours to get a PCR signal. And this is great because we can do all of this on site. We don't have to worry about shipping or anything like that. And our extraction process drastically reduces degradation after we get maybe halfway through it before we throw it in a PCR plate. Another part of this project was generating a Power BI dashboard where we could compare our wastewater signal to more clinical-based data in those communities like inpatient COVID beds or epidemiologic curves through some of those no swabs that we all hate. And the way that we were able to do this was through a new genome extraction process that we've used that's termed exclusion-based sample preparation, which is a relatively simple paramagnetic bead process that you might be familiar with, but it just uses a plastic plate that's reusable to limit some of the on-hand steps. For PCR, we have a sufficient amount of salt carryover to where we don't see too much degradation, but for sequencing, we need to do some other steps such as filtration or an additional impure bead cleanup to wash out some of those salts and things like that, and to bring our concentration up to a more sequenceable level. What's great for a low resource community is that this drastically reduces things like single-use pipette tip usage and is much quicker compared to other methods that use a similar process. And it's a lot easier. We've even piloted this process with, undergraduate trained students or wastewater treatment plant operators. And after a couple of runs with it, they're pretty comfortable with doing it as well. So here's a quick video on how it works. It's really just two magnets that sort of push these our sample coated beads through different wash buffers. And we're able to transfer these into different wash buffers and an elution buffer without a single pipette, really. And this is great because the cost of this is much lower per sample. And instead of having to fiddle around with a multi-channel pipette, we can just use a linear actuated motion. And our final elution is much more stable at room temperature. So we're able to sort of throw it in a van and drive back to our centralized lab for additional testing with no worries. So AMR was the next big target that we came up with that we're really excited about. Here along the Appalachian region of the US, we often have a prescription rate of over 1 prescription per person. There's a lot of reasons for that, lack of healthcare access and other things, but especially after the SARS-CoV-2 pandemic, where things like Z-Pak or amoxicillin were sort of handed out like candy, we really expect this to be even more of an issue in the coming years. So we first set out to gather some data on this using an Illumina platform kit, an RPIP, respiratory pathogen ID AMR enrichment panel, which is a bit of a mouthful, but it's an enrichment panel for often thousands of AMR genes and viruses and pathogenic bacteria and the such. So we have a great research hospital on campus that we could sort of contract out for this, but it's definitely at a price point that wouldn't be useful for a longer term study, at least in our lab. We were very excited with the data, though. We saw often AMR biomarkers related to almost every drug class that's tested on this panel. It is a clinical tool, so that we're sort of repurposing for wastewater purposes. So a lot of the data analysis is proprietary for obvious reasons. And so we weren't able to see exactly how it was calling certain things over other things. So we were a bit discouraged by that. But our clinical partners were very excited to see certain AMR genes that aren't routinely tested, that they were very interested to see that could be useful for policy making within the hospital. And so the next obvious choice was to use some lower cost platforms, such as the Oxford Nanopore platform, to sort of bring down the price point to a level that is conducive to research in Eastern Kentucky. We ran into a couple issues up front. Our process does involve some fragmentation and things like that. So things like an additional AMPER-B cleanup that I mentioned, or even duplex or high accuracy models really improved our reads. We often improved our quality score by up to 5 just by throwing it through a duplex. And for most of our runs, we were using the fungal flow cells and a couple of the large amino flow cells with the barcoding kits to, again, reduce some of that cost. And so AMR is a fire hydrant of data. This is just some of the stuff that we've collected. Vibrio cholera is not necessarily an ongoing outbreak in Eastern Kentucky, but it is an area that's prone to flooding and tornadoes and the such. So we are interested in sort of the resistome of that region for when things like that happen. So we had a lot of hits for genes like SOL1 and SOL2 with decent coverage, and for other Vibrio species that are known to be things like food poisoning and stuff like that. For a lot of this data, we do use epidemi labs, but we are slowly crawling towards more offline options as we try to accommodate some of our Central African partners who don't have consistent internet coverage. And so next, after Eastern Kentucky, we're going global with a great partnership with Arizona and the CAPAO that we've organized, where we are going to set up a lot of the sequencing centers in areas of the world where they're needed most. So the locations we've picked are places that are more prone to future pandemics where they might emerge, or for natural disasters as well. And so a lot of the issues that we've overcome in Eastern Kentucky with figuring out an on-site extraction process that's simple and cheap and low form factor to sort of ship out to these places is going to come in handy. And we're working with great partnerships, especially in Uganda, which is where I'll be hopefully in between recording this and seeing you in London. So hopefully it all went okay, where we'll do a pilot study and set up and train some great researchers there to do a similar style of work. And so this is where you come in. We really need your help. We're not a bioinformatics lab by any means, but we are using lots of sort of very standard data analysis tools like Fly or Praca, but we're very excited to see some of the newer AMR data set analysis tools that are coming out, and hopefully finding a way to use them with as little online connectivity as possible. So a lot of our partnerships are very adamant on doing the data analysis themselves. They really don't want to ship off their data to some guy in Kentucky and then never see it again, which is completely understandable. They care about their community and want to play a part in that as well, not just the sequencing side of things. And we're very excited for the future of the sort of the direct RNA or cDNA sequencing kits. Things like influenza and measles are very important to a lot of our partnerships, and the additional cost in a couple PCR steps or amplification stuff like that is somewhat inhibitory to some of our goals there. Things like PCR plates are worth their weight in gold, so very excited to see those. And I just wanted to thank especially my lab for supporting me through this, and for our University of Kentucky for funding this work and the CDC as well. So thank you so much.